Hi, how are you?
We're good. We're excited to be part of this today.
We're excited to have you. Thanks.
Hello.
Hello, everyone. Welcome to STROLO University. Today is Friday, May 14th, and we are in week 5 of our spring semester for STROLO University, and very excited to share with you a really mind-blowing presentation today. We have members from Translate Bio who are going to share with us some of the developing research around messenger RNA, which has been more commonly in the news in the past few months, more relating to COVID vaccine. But the STROLO University is a program offered by the Cystic Fibrosis Lifestyle Foundation, an organization where our mission is to help promote healthy and active lifestyles for the cystic fibrosis community to live their best lives in living stronger and longer, or STROLO. The CFLF was started in 2003 for exercise and recreation since 2007, and has since awarded more than 1,600 recreation grants and more than $800,000. And with the COVID shutdown and activities being suspended for a period of time, we grew into this educational space and providing online activities and group exercises, and now educational opportunities for mental empowerment and mental well-being in maintaining control of our CF. I am also an adult with cystic fibrosis. Today I'm turning 45, so halfway to 90. But so I just want to welcome everyone and encourage you to please utilize the chat window if you're watching through Zoom today, and if you are watching through Facebook, please use the comments throughout the presentation. We will be monitoring both chat windows, so we do welcome questions and participation. There will be a period at the end to address questions and discussion. So I'd like to introduce Jim Sullivan and Richard Wooster from Translate Bio, as well as my contact, Maura. I always mess up your last name.
Gavigan. That's okay, Brian. It's a tough one.
That's all right. You can mess up my name too.
Well, happy birthday, Brian. Thanks so much for having us today.
Thank you. Thank you. Really excited to have you guys. And I'll turn it over to you to start our presentation for today.
Absolutely. Just before I turn it over to the real stars of the show, Richard and Jim, I just want to say thank you for having us. It's been a pleasure to work with CF Lifestyle. Over the last few years, we had the pleasure of participating at the Knockout CF event back when in-person events were possible, and look forward to the return of that event in the future. But I'm excited to be here. I work on our corporate communications team and oversee our patient advocacy efforts. So if you have any questions after the presentation, please don't hesitate to get in touch with us through our social channels as well. But without further ado, I'll let Richard begin the presentation.
Wow, thank you, Maura, and thank you for the invitation today to come and share with you first some background, but then some data on the work that we're doing in terms of developing mRNA therapeutics for pulmonary diseases, with cystic fibrosis being really front and center for us, and our lead program, the lead disease, which we're actually working towards with our therapeutics. As Maura said, my name is Richard Wooster. I'm Chief Scientific Officer with Translate Bio. I've been with Translate for 2 years now. Before that, I've spent 15 years in the pharmaceutical industry, and before that in academic labs in London and Cambridge in the UK. Before I jump into the background, Jim, maybe I'll just ask you to introduce yourself so we can have a smooth transition once we get to that point.
Sure, yeah, thank you, Richard. So my name is Jim Sullivan. I started relatively recently here at Translate, and I'm really excited. I came in to head the pulmonary discovery effort. Before my before joining Translate, I spent a long time at Vertex. I worked at Vertex for about 12 years. And that was really represented a diversity of roles there that, at various times spanned really, really early research all the way through to late clinical development. And, even included some opportunities to be able to go out and have community-facing discussions about CF. And I always found it to be so rewarding in presenting data from Vertex and really thinking about the science and the translational science that we were doing, but then really to be able to understand how they were actually leveraged in the clinic and to be able to, talk to the people who were actually employing the medications, as well as an opportunity to meet some patients as well. So I had a brief period away from respiratory when I went and worked in cell and gene-based therapy, and I just had to get back to respiratory and CF. And so Richard presented the opportunity for me to come and join Translate. And I'm just really excited about the opportunity that Translate has now to take this platform that has been in development for a long, long time and really recognize the value that it could have for patients.
Fantastic, thanks, Jim. And so before jumping into some sort of real specifics, I think setting the stage for you in terms of mRNA and how we see the potential for this in terms of therapeutics. So Maura, if you could move to the next slide. I think many of you will be familiar with this paradigm where we inherit the DNA from our parents that encodes and how it contains our genetic information. And inside our cells, we have this process of transcription, which then reads the DNA to create the messenger RNA. And those instructions then move out into the part of the cell where proteins are made. And these proteins, not just the building blocks of our cells, but actually perform many, if not most, of the functions within our cells. And of course, these cells all build together to make us who we are. Of course, we know that there can be inherited defects in DNA, for example, in the CFTR gene in the case of cystic fibrosis. And so that sort of mutation, that defect in the DNA follows through this path, ultimately resulting in a defective protein, which then causes all the symptoms and the consequences of that defect in CFTR. Now, with a lot of existing small molecule drugs like Trikafta, really they're focused around the protein and how those small molecules combined interact with the proteins and change, modulate, if you like, the function of the protein. And that's fantastic if there's an opportunity for the protein to be made, or if there's protein, but it's not quite fully functional, fully effective. But when there's no protein, those molecules have nothing to bind to, nothing to work with. And so for those individuals with cystic fibrosis who are making no CFTR protein, really, we know that the modulators in most cases have really no effect. So what we've set out to do is to hit the center, hit the bullseye here in the middle of this slide with the mRNA. And so if we can deliver the functional mRNA, that can be translated and make the protein and restore that CFTR function. Now, you heard earlier that this sort of mRNA has come in perhaps into many people's sort of lexicon and understanding now because of vaccines. And let's think just briefly, I'll touch on vaccines in a few minutes again, but many of the existing vaccine technologies were using protein. And so a protein would be made in a manufacturing facility, and that's what would be injected into your arm or injected, and that's what would then cause that vaccine to be effective. Now, what we've seen over the past year, of course, is that mRNA vaccines for COVID-19 have been made, and now many tens, if not hundreds of millions of people have actually received an mRNA-based vaccine. And for us, I think an aspiration is to follow that and to have mRNA therapeutics really grow in the same way and be, I think, a mainstay of therapeutics in the same way that mRNA vaccines have done that in the infectious disease space. So how do we do this? And what are the components that can make an mRNA therapeutic? And that's shown on the next slide. So there are two building blocks that go into making our MRT platform. The first of those builds on that last slide. It's the mRNA itself. And there are these different sections of the mRNA which are critical to have an mRNA which is then going to create the protein. mRNA is a very unstable molecule. It can be degraded very rapidly. So we protect the mRNA in the structural sense by this 5' cap and the poly-A tail. And that protects that coding region in the middle. That's what codes for the CFTR protein. But even with those protections and with that mRNA, we need to deliver it to the cell where it's going to be expressed in the lung. And the way we do that is we package the mRNA inside a lipid nanoparticle. So here we've made these proprietary lipids that, when we mix them together with the mRNA, form this sphere, this ball-like structure. And what we do then is we put this into a nebulizer as a solution. That vibrating mesh nebulizer then creates an aerosol. That's what we're looking for patients to inhale. And then that lipid nanoparticle takes the mRNA through into the lungs and into the cells to make protein. So to show you how all of that comes together, we have a short animation, which is on the next slide.
Deep within cells, genetic information encoded within our DNA is transcribed into messenger RNA, or mRNA, which is needed to produce proteins, the molecules that perform essential functions in our bodies. mRNA sends a message to our cells with instructions to produce proteins through a process known as translation. Genetic diseases are caused by mutations in DNA sequences that result in mRNA providing the wrong instructions to cells. This leads to the production of dysfunctional proteins, or none at all. Translate Bio aims to treat diseases by addressing the central step of our body's protein production process. We design and optimize an mRNA sequence containing instructions for making the desired protein, and then package it into a customized delivery vehicle, the lipid nanoparticle. The lipid nanoparticle protects the mRNA from degradation until it reaches the target cells in the body, where it provides instructions to enable the cell's own machinery to make functional proteins. Translate Bio believes that mRNA technology can be applied broadly across many diseases where the production of a desired protein could provide therapeutic benefit. mRNA may also be used to make vaccines that prevent disease. In this case, the mRNA vaccine is intended to provide instructions for the cells to produce a viral antigen, causing the body to generate a protective immune response. Relying on more than a decade of mRNA expertise, our unique platform of mRNA design and delivery is at the core of our mission. Developing transformative mRNA medicines to treat or prevent diseases.
Oh, Richard, I think you're muted.
Sorry, I was muted there while the animation. This is a static version of that animation, just so you can recap on the different steps here. And as you can see, as the LNP is taken up by the cell, what we do in the labs is we actually optimize the different components here. So we make different lipids, we test different mRNAs, really to drive ultimately the protein expression, which you can see on the right-hand side of here with that ribosome, which is taking the mRNA that we've made and producing these proteins. And so we've built now a team who have the expertise to go through those different stages of optimizing the molecules. We test them in our preclinical models to assess whether they're effective or not. And that, of course, is an important step before we ask patients to work with us and actually start to evaluate these in clinical studies. We mentioned vaccines as well in that animation. And there are some important differences between what we do for our pulmonary applications compared to vaccines. Of course, in a vaccine setting, we're looking to make the antigen, for example, for the coronavirus. Whereas for cystic fibrosis, the protein is that natural protein that is not present in many CF patients, CFTR. And of course, with a vaccine, that's an intramuscular administration, whereas we have now built a strong group in terms of this inhaled approach to administering our LNPs. And with a vaccine, it might be there's only 1-2 doses. Perhaps for life, we'll see what happens with the coronavirus, whether there needs to be annual boosters. But for cystic fibrosis and other chronic disease settings, we're expecting that these would be taken on a routine, regular basis by individuals to help restore, in the case of cystic fibrosis, the lung function. Now, you might be asking the question, well, how does mRNA compare to other types of genetic modalities? Because there are certainly other opportunities. And so what this slide is showing is that with mRNA, we're absolutely using the cell's machinery to make the protein. And if you could advance Maura to sort of, to build out the slide. So we're making the cell's own protein with mRNA. Because gene editing, I think that's a real sort of a goal, which certainly is would be a great place to get to. We're still working through, I would say, in terms of how to do that and other aspects of where that could take us to. But with gene editing, you're making that change, and then if that's there, the change is made and can be undone, can be altered. With mRNA, what we see is we can change the dose, we can change the frequency, we can really work with patients on what's best and what's going to give them the best outcome. And so we're excited about mRNA therapeutics for now and where they're taking us. And what I'll do is hand over to Jim to describe from this sort of theoretical application of mRNA now into real data and a real practical application.
That's great, Richard. Thank you so much. Mara, if you want to advance the slide, I just want to actually begin by reinforcing, some comments that Richard made. It particularly where, we're all really excited here about, the fact that we have demonstrated, or that the community has demonstrated, the utility of mRNA-based vaccines. It's really exciting, but at the same time, that's obviously a different context than of thinking about mRNA as a therapeutic for a disease that is a chronic genetic disease. And so let's just take a step back. I know everybody here is aware, but CF is a monogenic disease. And so really what it means is that patients that have CF, have are homozygous for mutations within the CFTR gene, right? The gene that is producing the CFTR protein that's responsible for ion transport, specifically, in particular, chloride transport across the epithelium, and where the deficiency of CFTR in the lung is essentially the underlying cause of the following disease pathology that results from this genetic disease. But it's also worth pointing out at this point in time that, this term monogenic means one gene. And there's an amazing amount of complexity that underlies that, which at this point, I think everybody in the CF community is well aware of, given the fact that the different CFTR modulators have different indications based upon the particular mutational profile. So just to highlight that. And really, as we think about that and we advance to the next slide, Mara, when we think about what the goal of the promise of these mRNA therapeutics are, one of the key promises is the fact that it has the potential to reach all patients, regardless of the underlying mutation. And again, as Richard points out, that also includes patients that have mutations for which a CFTR modulator would not be a viable mechanism because of the fact that little or no protein may be produced. And really the opportunity then to deliver the mRNA really, as Richard also pointed out, means that it's an opportunity to treat the underlying cause of the disease rather than treating symptoms, downstream consequences of not having a functional CFTR protein. And so if we take a dive a little further and think about what that first line means in terms of the diversity of genetic mutations that can cause CF, this is a schema that I'm sure many of you are familiar with, where essentially what we're looking at now are the different classes of CF causing mutations, the different types of CFTR deficiencies that can present and cause CF. In these different classes, the CF research community has done a great job at spending a lot of time characterizing these different classes of mutation, understanding the distinction between a protein defect like Delta 508, F508 del, which is this class 2 defect that results in a folding defect or trafficking defect, as compared with a mutation like G551D, the classical kaleido responsive mutation, where a protein is made and is able to make it to the cell surface, but is non-functional. And essentially, regardless of which class of mutation underlies the cause of disease, being able to have this therapeutic platform that's described on the left-hand side that involves delivery of a novel mRNA that can produce a full-length protein should affect the underlying cause of disease, regardless of which mutations a patient would have. And as Richard pointed out, that also does include this class 1 deficiency, where patients aren't making full-length CFTR protein. And when we start to think about how that relates to patient populations, and Richard already commented on this earlier, but if we kind of look at the distribution then of these different types, not necessarily on the next slide, Maura, of the different classes of mutation, but we now split these up into essentially therapeutic types, it really is this 9.4% of the population where a CFTR modulator is not indicated, won't provide benefit, where you could imagine, a really nice opportunity to be able to have this mRNA therapeutic platform that could essentially then replace the deficient CFTR protein, recognizing that there may also be some additional patients even that have mutations that are indicated for some of the CFTR modulators where patients may not be deriving benefit. So if we think about how this relates to our clinical application in the next slide, we do have, and if you want to advance one more slide, Maura. Yeah, perfect. So what we're going to do now is just kind of very briefly go over the translates first assessment of a CFTR mRNA therapeutic packaged in one of these lipid nanoparticles in a clinical trial. And really, the thing to point out here, one of the key aspects of this is really that this trial design reflected a first in human trial. And a key point there that I want to highlight is that the objective that's defined on the left-hand side is really to evaluate the safety and tolerability of this, particularly in CF patients. Can we actually deliver this LNP, this mRNA packaged within a lipid nanoparticle to the lungs of a CF patient, and will it be safe and tolerable? And in order to assess that, we looked at this in the context of a randomized double-blind placebo-controlled trial. And all that means is that, the investigators and patients didn't know whether they were getting a placebo or an active drug, so that we could make sure that the trial was run with all the proper integrity, and that the interpretations that we were drawing related to that objective of safety and tolerability were robust. And then one other piece that I want to comment on here for the results that we have reported so far that are defined in this blue box is that they incorporated both what we call SAD and MAD groups, where the groups on the left-hand side in part A reflect a single ascending dose, where patients were administered one dose at these different dose levels. And then ultimately, the safety and tolerability outcomes from those enabled the assessment of multiple ascending dose cohorts, where we're able to assess the safety and tolerability in the context of repeated doses up to 5 weekly doses. The last comment that I want to make about this particular clinical trial is that it's actually still ongoing. And so while we're talking about some some interim data based upon the shaded cohorts in parts A and B, that there are still cohorts that are ongoing that include a 20 milligram dose in a MAD cohort, as well as what's defined in purple, which is an effort in order to assess how daily dosing, the tolerability of daily dosing as compared with a weekly dosing regimen. And on the next slide, we're just going to highlight a really high-level overview of what our outcomes have been to date from this study. And just as a reminder, this study, again, was designed in order to assess the safety and tolerability. And as the first inhaled mRNA therapeutic, particularly, also the first to be administered via repeat dose, the main high-level finding so far from the interim analysis is that it has been generally safe and well tolerated. Lung function, which everybody here knows well, specifically percent predicted FEV1, was included as a safety endpoint. And to that end, it was not negatively impacted in those patients that were receiving the active treatment. But at the same time, there were no obvious patterns of increases in percent predicted FEV1 either. And then when we think about two other parameters that we were really interested in this study, the first one was really understanding whether or not the presence of the CFTR protein, which would be generated from the mRNA that was delivered, whether or not that or the delivery vehicle itself could be immunogenic. And we tested that, and the conclusion is that there are no clinically relevant immunogenicity by looking at both anti-CFTR antibodies. Are we generating immunogenicity to the protein that we're delivering, CFTR, or to part of the delivery vehicle, which is this anti-PEG antibody, as well as whether or not we see our body's immune response being activated to CFTR. And the high-level conclusion is that we're not seeing any of those patterns. And then the last piece is really, one of the key questions here, one of the key challenges, truthfully, which I think we all recognize as a community, is that when we're delivering a inhaled route of administration for any CF product, we don't have the opportunity to be looking at systemic biomarkers that could tell us what our levels of, whether or not we were hitting target levels of exposure, but also, as we're all used to in thinking about systemically delivered modulators, the opportunity to look at sweat chloride, for example, because we really are targeting delivery specifically to the lung. What we did decide to do, though, was to look in the blood to see whether or not we could detect any mRNA or lipid. And we did detect both mRNA and lipid sporadically, which suggests the fact that we are achieving mucus layer penetration with the therapeutic. And so if you advance to the next slide, Mara, these data, really put us in a position where, for one, we obviously want to make sure that we're completing our ongoing phase 1 and 2 trial, and really understanding everything we can learn from that, but also where we're really excited about being able, especially in the role that I've come in on as leading the pulmonary discovery effort, to be able to come in and really think about how we can even improve upon what we've developed so far. And so that really is what my team is focused on, is really thinking about what's our next generation platform that can really push us to an improved level of potency or an improved, administration profile. What can we do in order to really improve the probability that we're going to be able to develop a treatment that will have the type of efficacy that we're looking for? And what we've done, between Richard and myself, has really been to build out a pulmonary research capacity that actually includes all of these components, really making sure that we're understanding the biology, both from an in vitro, as well as an in vivo perspective, understanding, the cellular, but also the organismal. And recognizing that with this complex drug product, we also need to really become even better at understanding, things like aerosol science and being able to understand how we can optimize the delivery of this platform. So with that, Mara, we just have a couple of additional slides that are starting to provide a view of how we're thinking about what those next generation assets could look like. And when, Richard had, already commented nicely that these mRNA products really consist of two pieces. One is the mRNA, the other is the delivery vehicle. And we're looking at both of those and what we can do to really improve the potency for each one. And what's shown here on the right-hand side is a chloride current from an Using chamber assay that I'm sure many people here will be familiar with. And what's shown in this purplish blue line in this Using chamber current is really looking at our original codon optimized sequence and seeing what tricks can we pull in terms of coding the exact same amino acid sequence, but actually changing the underlying mRNA. Let's make wild type CFTR protein, but let's figure out if we can optimize the sequence that creates that. And in doing so, what we can see is a 1.5-fold increase from some early, assessments of some of these codon optimized variants. So now these are still encoding the exact same wild type amino acid sequence, but getting there via a different mRNA solution that will allow for increased amount of protein, increased amount of CFTR function for the amount of material that's delivered. And what we're looking at specifically within this plot are two different ways of stimulating CFTR that's shown in these first two arrows. And then subsequently in the last arrow, really confirming with utilization of a CFTR inhibitor that this change that we're getting is driven through CFTR function. So we'll continue to be moving these forward and assessing ways of improving the level of CFTR function that we can get by optimizing that payload component of our drug treatment. The other component that we've already spoken to, we'll cover here on the next slide, which is our last piece of data, is really thinking about doing everything we can in order to increase the level of, the potency of the lipids that we're utilizing when we're administering the RNA payload. And what's shown here is a method called immunofluorescence. What we're looking at is we're looking at microscopy images from a mouse, where essentially we're looking at epithelial cells and where this DAPI stain is staining for a nucleus. So every time you see one of these little blue dots, that reflects the nucleus of a cell. And what we're looking at in this orange is specifically the expression of a CFTR protein. And so what we're comparing in this slide between the top and bottom rows is either a saline control, where we're essentially just administering saline to animals, and where we can see our DAPI stain, everything looks fine, we can see the nuclei, but yet we don't see any CFTR expression. And when we deliver a lead lipid for NextGen, encoding our CFTR mRNA, we can see not only with this middle panel that's detecting CFTR in the appropriate localization, that actually when we do that DAPI CFTR overlay, and particularly in the blow up on the right hand side, we can actually see that the CFTR is really localized in the apical epithelium, the top of these epithelial cells, exactly where we would expect CFTR to be expressing in its normal endogenous form, where it's able to be present on the top membrane, this apical membrane, and be able to carry out its intended function of transporting ions that will then in turn drive the appropriate hydration of the mucus and allow theoretically restoration of the cilia function, and in turn, essentially limit that pathophysiological cascade that results from CFTR dysfunction. So this is just a little flavor of the type of work that we're doing. And, it's a question not only of, continuing to advance this research so that we can better understand and better characterize additional delivery vehicles and additional cargoes, but also increasing our depth of knowledge by doing assays like this and some additional assays that really help us to understand, how to be administering these compounds, down the road as we advance them towards the clinic. So, it's really exciting for us. I hope that you can feel that, and I hope that you guys share some of that excitement. It certainly has been fun to be able to have an opportunity to speak to this group, and I'm sure I'm speaking on both Richard and my behalf when I say that. And certainly, if there are any questions, we'd welcome them.
Thank you so much, Jim and Richard. That really, at least for me, easy to follow. A couple questions I had was for the delivery of this type of treatment through nebulization. I think you touched upon this, but is it still being looked at in terms of if it's requiring particular droplet size and distribution and special nebulization technology, or if it would be, a little more, I should say, generic in terms of nebulization?
Yeah, it's Brian, correct? Is that right?
Correct. Yep.
Brian, it's such a great question. And it suggests that maybe we should have had the conversation focused at an entirely different level, because it's actually like a really probably not a very intuitive question when you're first thinking about this. You're inhaling something, you're delivering it to the lung. It's as simple as that, right? And your question, really belies the fact that it's not that simple. And so that really is part of the build out of really making sure that we understand the aerosol sciences, that we're characterizing for this particular delivery vehicle when we're making it in this particular way, what the robustness of development of those appropriately sized particles are that will, with the appropriate combination of device and drug, make it to the right place. And so that all has to be part of that research pipeline, Brian. So thank you for that question.
Thank you. And we have another question in the chat. Are you guys able to see the chat, or do you want me to read it to you? Great.
Yeah, so maybe Juan, thank you for the question. Maybe I'll take a stab at it, and then Richard, see if you have any additional comments related to it. So you're right. And again, this is also a really informed question, in that CFTR is big, and that also means that CFTR can't be efficiently packaged across any delivery vehicle that's out there in the drug development world, right? Specifically, as we think about LNPs, the size and structure of the LNPs is certainly sufficient to package CFTR mRNA. So it's a good question, but in part, some of those preclinical data that we were just looking at, those were delivered. The CFTR that we were looking at in those immunofluorescence images were delivered by LNP. And ultimately, there's a lot of other research that, will go into that for, really confirming the fact that we can package and express to the degree that, we ultimately would need to. Richard, do you have any additional comments related to packing efficiency?
No, I think it is a great question, because I'm following up here, the CFTR mRNA is about 5 kilobases in length, which makes it, I don't know, maybe 3-4 fold larger than the average human gene. So it's definitely, sort of on that larger side. What we've done over time is actually figured out the manufacturing approach to be able to successfully make that long mRNA to the quality that you'd want for a drug to actually asking people to take. So we've got that in terms of the size. And then our lipid nanoparticles, we aim for about 100 nanometer diameter. So these are really small in terms of size. And then we know from where we've looked at these, we can get multiple copies, multiple molecules of mRNA for CFTR into each of those lipid nanoparticles. So we do know we can load them up with a number of different mRNA molecules. And so those are then going to get nebulized, go down into the lungs, and to be taken up by the cells. And I think that part, maybe I just add a bit about the nebulization. So, of course, it's known that this vibrating mesh that's inside the nebulizer, so essentially it's a piece of metal with laser drilled holes in it, which vibrates very quickly. And depending on the size of the holes and the shape of the holes, the droplets that come out can be different sizes. And so we absolutely look at the size range of droplets that are going to get down into the appropriate areas of a human lung to be able to do that. And so as we make those droplets, we can characterize their size and the distribution of those to make sure the nebulizer is giving us what we need. And then we have the analytical methods for looking at the LNPs, the mRNA, all of those different components. Jim, maybe I'll hand back to you for the FEV1 question.
Yeah, so Isabel, it's a great question, recognizing that both the variability in FEV1 as well as the fact that there's real biological patients' FEV1 is changing all the time, aside from the fact that there's variability in the measure to begin with. And so I think it's a great question, but it's not what the study was designed for. And it's certainly, especially while the study is ongoing, all that we could say about it right now is just really what we know from that interim analysis, which is that there's no pattern of change based on FEV1. But I share your curiosity, Isabel.
Thank you, sir.
And Jim, do you want to cover sort of, as you were talking about, delivery to specific cells? Are we only seeing delivery to epithelial cells? Are we targeting other data that we've seen?
Yeah, so it's also another great question, and it's part of what we will be characterizing moving forward to really understand what that distribution looks like. I can tell you that is where one of the potential concerns you would think about in a so what I'm trying to figure out what the best way to answer this question is. So I'm sorry that I'm going back and forth a little bit, but what I will say is that it will continue to be part of our characterization in order to understand which cell types we're targeting, not just in terms of epithelial versus non-epithelial, but also epithelial in terms of different types of epithelial cells. But what I will say is that we're primarily targeting epithelial as compared with non-epithelial cells.
And then in terms of the potential effectiveness in the smaller or larger airways, I think from what we've seen, Jim's last slide showing that expression of the CFTR protein, which we've seen in our animal studies, I think we have achieved expression across a wide number of different areas within the lungs. And so I think we are looking to get potentially that effective level of expression in both the smaller and the larger airways, really that how we can get the maximum benefit from delivering the mRNA.
Another question I had was, I'm making the assumption that this type of therapy is an ongoing therapy. It's not a conclusive solution to CF, but Are there primary risks that are considered likely, or in terms of tolerance by other organs? Is it something that's manufactured through the liver or the kidneys? And I guess how manageable for the body overall have you seen it?
Maybe I'll start, and then Jim, you could pick it up. Because the route of administration is inhaled, what we've seen as we've measured in our sort of preclinical studies, but also in collecting blood samples during our clinical study, we see very small amounts of the mRNA or the LNP actually elsewhere in the body. It really is sort of heavily concentrated within the lungs. Because that's the part of the sort of all of the different aspects of CF. This is really looking to address that lung component of the disease. So certainly from that, but Jim, maybe I'll pass to you and other thoughts.
Yeah, Richard, I don't have anything to add. I
Okay.
mean, that was the exact same thought. And as we think about moving these things forward, Brian, that's part of, our evaluation is, like, let's understand what kind of systemic exposure we have. Are there, other properties of these drugs that we need to think about beyond just, a question of transfection.
And just overall, it may have been addressed in the presentation that I just didn't catch, but in layman terms, what would you say has been your biggest struggle or biggest challenge to overcome in these studies of such new type of technology to address? And again, as we've talked about the mechanisms of mRNA through vaccine being different than that of inhaled mRNA. Just curious what you found to be the biggest.
So maybe I'll give one example, which I think around the whole mRNA platform side, and then Jim, maybe you could talk specifically about CFTR. As Jim showed you, the clinical trial for that we've done, the highest dose that was given in each dose level, or at the highest dose level, was 24 milligrams of drug. Now you compare that, if you look at the mRNA vaccines, if you're actually to go sort of go on and find the dose level, it's usually in the maybe tens of micrograms. So what that sort of is saying is a really big difference. But what we've done, we set out really from the earliest days, was to have the manufacturing to be able to make that large amount of material. Because not only is each dose, maybe 1,000-fold higher than you would give for a vaccine, but potentially people are taking this every week. And so we've set out that was a challenge which was recognized now many years ago for us, but we've been working and now can make these large amounts of the mRNA. So certainly that's a challenge which we've solved for. Jim, in terms of the CFTR, you've been working in the CFTR field for many years now, so you've seen, no doubt, many challenges.
Yeah. It's such a interesting question, Brian. And, I think the first one, I see two major challenges. And the first one is the obvious one for everybody here in this call, which is that CFTR was identified 32 years ago, right? It was identified in 1989. And there was a view that now that we know what the gene is, we can just move straight into thinking about exactly what we're talking about now. We can think about, gene therapy, right? Or, mRNA therapy wasn't, in the picture at that point. But the overall view that having identified the gene means that we're just around the corner from being able to deliver a therapeutic from that, is as compared with the reality that we've had to live through over the course of the past 30 years, right? And so delivery far and away is the number one challenge for the field, for the community. Richard displayed a number of, mechanisms. He talked about mRNA, he talked about gene editing, And across the board, I think we all recognize that being able to deliver the material, whatever that material is, is the key challenge. And that's part of the reason why we've invested what we have into really optimizing our LNP delivery platforms and understanding the components that you already asked about earlier, Brian, which are the components that go beyond the actual chemistry of those LNPs, but actually understanding how we can actually get this delivered. So, I think I'm probably speaking less from, my experience, my recent experience here at Translate, and more kind of reflecting probably the way that I think this community has been thinking about it. So that's one. And then the second piece that I would give is actually now specific for thinking about, for example, the clinical trial that we just defined, where we actually, took the really nice step, really, really important step of being able to assess safety and tolerability of a repeat dose administration of an LNP. But, Isabel's asking about other endpoints, how patients feel, those kinds of things. And I will say that, one of the other challenges that we have as a community of thinking about pulmonary-based delivery approaches is that pharmacodynamic endpoints are hard to come by. And so, again, I think we all as a community wish we were in the situation where we had a biomarker like sweat chloride, where we could just, look and see whether or not we're actually having, a clinical impact, even from a relatively small cohort of patients. And so that's just, a challenge that is inherent within, the pulmonary delivery space that, again, we're just as a community going to need to work through. So those are my top two, Brian. And then it's a big drop off beyond those two.
And just a last question I have is the intention of this type of treatment, or I guess even necessity of this treatment, mutually exclusive from CFTR modulators, or is it something that potentially might be used in conjunction with?
Jim,
yeah, sure. Yeah, it's a great question. And, certainly, if where the protein, that we displayed on that last Using chamber slide, is wild type CFTR. And so, in the context of a CFTR potentiator that could potentiate wild type CFTR, you could certainly, it's not only logical, but it's the appropriate inference to make that, if you're delivering wild type CFTR, and then you also have a CFTR potentiator that is interacting with that wild type protein that's known to potentiate that particular protein, that there could be a additive, that they could kind of act together, so to speak, to increase the amount of, increase the open probability, increase the amount of ion transport. It's a perfectly reasonable supposition, Brian. Yeah.
Wow, this is amazing. So I just want to thank you guys again. This has really been such an in-depth and understandable presentation. And I do want to thank you again for your time and contribution to the STROLO University series. And yeah, I just am honored. I'm still kind of digesting everything you just shared. So I'm really, really excited about these prospects. And Maura, I think you've known from the first day we met a couple of years ago that, how excited I've been, especially for that population of the CF community that is not able to benefit from CFTR modulators. So thank you for the hard work that you're doing and the cutting edge thinking that you're doing. You guys truly are heroes.
Thanks so much for having us, Brian. It was really, we're humbled, to be part of this series and to be working with you. And we always say how fortunate we feel to be part of this community. There's really nothing like the CF community and the commitment from the whole community to research and novel treatments like what we're doing is, just a huge part of why we do what we do every day. So thrilled to be part of this. And like I said at the start of the call, we are, open to any questions that come up based on this. We're very active on our social channels, and we actually just launched our CF Month campaign where you can learn even more about what we're doing and hear from Richard and Jim as part of that series as well. So I encourage everyone to head over to our channels and follow along, and that's where we share all of our updates and progress as well. So that's the best place to kind of keep up to date on all the things going on at Translate Bio.
Absolutely, and hopefully we can also revisit this in our fall semester. So that is something that is ongoing for us, and we look forward to some of the updates as well. And I do have a special treat for viewers. We have started a Watch to Win series. So in just a moment, I will share with everyone our letter for today. It's a matter of collecting letters each class during the week and putting those words together. And this week, the first person that is able to put together the word and email it to us as of 1 o'clock on Sunday afternoon Eastern Time will win a $50 gift card to the Amazon. Sorry, brain is shutting down. But also wanted to just do a quick shout out again to our supportive sponsors, Chiesi, Viatris, Nestle Health Science winners today, and Kroger Health as well. So really could not do this program without the support of all of our sponsors, and just really grateful for such support again. But I am going to stop the live Facebook stream and then share with everyone the letter for today. But for those of you on Facebook, tune in live to our next series is at 6 o'clock today. And we have Colton Underwood from the Legacy Foundation, along with his partners at AffloVest. So 6 o'clock, please join us back. And for those on the call, please hang on 1 second while I stop the live stream. Okay, and for everyone on the call today, the letter for this class is the letter T. So that is the fourth letter of this week that we've put. Is that right, Tiff? That's our fourth letter. So we're getting there. We'll have one more letter today, 2 on Saturday and 2 on Sunday, and you will have enough letters to solve the word. And I'll give you a hint that it's a word that I have very much of for Jim and Richard and Maura today.
Well, thank you so much, Brian. And we're excited to hopefully be part of the fall semester as well. So hopefully this is to be continued.
Great.
Thanks,
Sounds good.
Thanks so
Thank you
much.
all again. Have a great rest of your day.
Yeah.
Happy
Thanks. Take
birthday
care.
again, Brian.
Take care.
Enjoy the day. Bye-bye.
Bye.